And so we pick up on this part of your packet where we discuss the association areas working together. In the previous slide, we saw that there in the lobes that there are regions such as occipital lobe, and it will contain the primary visual cortex, areas that receive information, and then it also contains visual association area, and that means that certain parts of it interpret what you are seeing. And you can do this for absolutely every single one of your special senses. You can do it for vision, occipital lobe, hearing, temporal lobe. You can do it for taste and smell in your insula lobe. And again, they have two parts to them, a part that receives information and an association area which interprets area. And they all, in an essence, have that very thing. Two parts that receive the sense, parts that interpret the sense. But now, as we look at this photo, when we use this photo, we used it to talk about the ventricles. And there's a structure to be aware of that comes up in lab called the corpus callosum. And this corpus callosum, what it is, it's a bunch of axons connecting to both hemispheres of the brain. As you look at this part of the brain, you'll see that there is a right hemisphere and a left hemisphere. And they have to communicate with each other through this corpus callosum. And this is the same structure that you see from a sagittal view right here, this larger white object here. And what the corpus callosum does is that it allows, or it coordinates signals between the two lobes, the two hemispheres, I should say, not the two lobes. Hemispheres is more appropriate. And I can't cross it out, so hemispheres. And as we move into this next area, what this is about, this is called integrative centers. And what this means is that you receive information from different association areas. Again, it's like if you're comboing visual information with auditory information, right? When you're using those two senses at the same time, your brain has to share or sort of share what each one knows. Share what it knows in the visual association area and the auditory visual information. And so it can even link up what you hear with what you do with your mouth. It can link up what you say with what you see. And so you can see it can even direct motor activities. And so it's you can see that it's going to be when you use a combination of different association areas. An example, when we look at our first one, Broca's area, this is going to be what we consider a motor speech area. And I want you to think about what do you need to speak as a human being? You need to be able to regulate muscles in breathing, muscles around your larynx to control what you say. But at the same time, you need to be able to interpret and sort of think of the words, say them in your head. And so then once you have the words that you want to say, that you've learned, then you use your motor components of your frontal lobe to talk. And so Broca's area, this little region here, this region is known that if you damage this region, you kind of might damage your ability to produce speech. You can make sounds, but not form words. So you might even be able to think clearly, but you will not be able to speak clearly. And so that's because you need to use motor. Remember that your frontal lobe possesses your motor aspect of your brain. And then temporal. When you hear words that you've heard and learned a language, you are using this part of your brain. And so you can see why this is going to be sort of a combo of association area from temporal lobe and areas that work in moving muscles from the frontal lobe. And that's what these integrative centers are. First one is Broca's area. This will be responsible in speech production. The prefrontal cortex is going to be a region that incorporates a bit of sensory information. And this is what helps us sort of make or use our critical thinking, think. It allows us to take in what we see, to hear, and then put it together. And so you can see as you see this PowerPoint, as you hear my voice, you are using vision and auditory senses to figure out what in the world I'm talking about, what in the world these slides are saying, what's the take home. Your learning regions are there. So we can put perform intellectual functions, and then we can put such as learning and problem solving. Usually most things can be summed up in learning, problem solving. You have a frontal eye region, and again, the frontal lobe is responsible for motor aspects. So even the muscles around your eye sends impulses eventually to the occipital lobe, but the muscles around it are controlled by the frontal lobe, which controls your motor cortex, your primary motor cortex, where you control muscles. And so your frontal eye field controls eye movements. And lastly, we have the Wernicke's area. This is your ability to understand auditory language. Like here, language comprehension. Your ability to understand language. Wernicke's area. All right. And so we make our way to our senses. And now, what exactly are senses? We're going to go over our special senses. And remember that this includes olfaction, taste, vision, balance, and hearing. You don't see touch. You don't see pain. You don't see temperature. The word senses, again, means touch, pressure, pain, temperature, and special senses are going to be these 5 that you are seeing here. So with that, we start off with smell, olfaction. And what we see inside our nasal cavity on the very surface, you can see a bone that looks very familiar if we look here. This is our cribriform plate. And remember those little holes that came out of that cribriform plate called the cribriform foramina? Those allow these olfactory extensions, these nerves, to exit. Cranial nerve number 1, which is going to be your olfactory nerve. And you can see once it arrives to the roof of that cribriform plate, it's going to send a lot of extensions and nerves through the bone, and that's what we're seeing here. You are seeing that on the epithelial lining of this nasal cavity, you encounter smell receptors that we simply call olfactory receptor cells. And olfactory receptor cells are going to be responsible for the ability to pick up what we call odorants. And I'm actually not seeing the word here. Odorants, like I said, I didn't put it in. And they pick up odorants. I'm going to write it in. Detect odorants. Like odors, odorants. And let me reposition my iPad here. There will also be cells called supporting cells and basal cells. And basal cells' main job is to create new olfactory epithelium. They're the stem cells of our receptor cells. And these are one of the only places that there is actual neuronal replacement. And so using our photo, olfactory receptor cell, and then our basal cell. Our basal cells, these will become eventually divide to replace worn out receptor cells. And so the pathway that things take when you smell things, as you smell them, all those receptors are located in the epithelial lining of the nasal cavity. You smell them, you see that the nerves are going to send action potentials, nerve impulses, into this olfactory bulb, and olfactory bulb is going to be our first cranial nerve number 1. It's going to send all those action potentials to the relay center, and then we'll leave it at that. And then so olfactory nerve handles smells, and that's all there is. The last thing to add is this lamina propria. As you can see, this is going to be the connective tissue layer that's underneath the mucosa, underneath the epithelium. Connective tissue layer, lamina propria. And so, only thing to show is that you can see that there's this gland here. This will secrete mucus, and this mucus will help all those molecules dissolve into the space, and then those action potentials will be sent into the brain by all these olfactory receptors. All right. And so now we move on to taste. Your tongue is this receptor machine in a way. It's meant to pick up molecules. And so what we see is that it is completely not just random taste buds spread out, that you have very particular taste buds, and they are like in certain regions. For example, as you look at this region, you can see that this is where you taste sweet, salty, bitter, sour, and there's another taste called umami, which is right here, and this is where it's detecting that flavor. And then lastly, you have receptors almost near the base of your tongue. Here you can see the epiglottis that protects the larynx. This is where you contain water receptors, and this is kind of where you kind of develop that desire, that taste for water, right? That you know you're drinking water receptors. And so these regions have what we call taste buds, but the taste buds are organized in unique little crevices. Like, for example, here's taste buds, but they are in these little tiny little crevices. These are called valle papillae, because you'll notice that they are all forming a V. And so that's why these are called valle papillae, because they are all arranged. And you can see these when you look in the mirror and you look back at the base of your tongue or the root of your tongue. You'll be seeing these very receptors there. And so we're going to zoom in out in the next page to see what exactly taste buds are. But you can see they organize themselves either there, or they will go to the foliate papillae, which are found at the edges of the tongue. And at the edges of the tongue, you can see that there are also taste buds in a unique crevices compared to the valle papillae. Very different. And then fungiform and filiform, you can see that the crevices look completely different. Fungiform is going to contain your taste buds in a different fashion. Filiform, the exact same thing. And the only thing that's different is not only their appearance, but where you find them. And so their appearances are slightly different. Learn the names of these papillae and the appearance you're seeing on this image. And the only thing to grab from here, aside from these 4 papillae, is that they contain the taste receptors. Which are also referred to as gustatory, right? Gustation. If you've ever taken Spanish, you've heard no me gusta, si te gusta, right? Si te gusta, gusta is like saying, oh, that is your taste. It isn't in your taste. Anything else to add? Now, these 4 papillae altogether are called lingual papillae, which in lingual in Latin means tongue. Papilla in Latin means nipple-shaped. We've seen this word before. Papillary layer, dermal papillae. Papilla means usually nipple-shaped, in a way. And so these lingual papillae, you have 4 types. And they contain your taste buds. The valet papillae, don't forget it forms that V at the back of the tongue. The foliate is on the sides, the lateral margins, which means the side edges in English. Fungiform are named after that because they look like fungal caps, like fundus, kind of, like where the word came from, fundus, like a fungal cap, like I kind of drew a little bad one here. But fungiform is where it comes from. It's sort of fungal shape, fungus. And then our filiform, these are particularly unique because they don't actually have any taste buds. That's what makes our filiform unique. You'll even notice in the photo, no taste buds highlighted. And what they do is that they provide friction. Oops, sorry. And that's all we got. So our taste buds, if we zoom in and look at what they actually look like, this is what we would be seeing. This is what it looks like in there. If you draw it, right, we zoom in one more time, you can expect to see little receptor regions here. So we zoom in on with a microscope, and this is what we see. We see this region, this region, this region, this region, this region. These are all taste buds. And so if we zoom in even more, this is what one individual taste bud looks like. And so what each one of these have is that as things are flowing, right, you have saliva up here and the food that you ate, like breakfast, then those little molecules from your food are going to make their way into these crevices, and then they are going to be exposed to the taste bud. And so that is arriving here. Like you're a little food molecule, you end up here, and now those molecules will be interpreted by these cells. And now from a field biological, and remember biology is a philosophy, it's called the natural philosophy, right? It sort of studies life, so why wouldn't it be a philosophy? Why don't you just taste the flavor food? Like, how come you can't just eat like a dog eats dog food every day and it doesn't know any better, right? Or maybe it does, but we don't know, right? But it doesn't know any better. It just knows it's hungry, it eats. How come as a human being, as a living thing, you watch any other animal and you don't see the necessity for them to flavor their fish, flavor their plankton, flavor the plants that they eat? They just eat it. To them, it's just like when they chew it, they're like, oh, this flavor is living flavor. It means I'm going to stay alive longer if I eat this. That's the only flavor that they kind of really know is staying alive flavor. If I eat this, it'll keep me alive. Human beings have been alive and migrated throughout the whole world. And so what this has benefited us is that flavor profiles of things usually tell you if they'll kill you. If something is poisonous in nature, if you're eating a berry, right, you get the slowest guy in the village to try out the stuff first, and then they try it out, and then they try it first, or they feed it to the dogs to see what happens, right? And then they eat it, and then They die or they live. And now the whole village knows, okay, this will keep us alive. And then, but usually he more than likely would say, the last words he was probably saying was, oh, wow, this is really bitter. And so that means he was eating poison. But then other groups of people, other populations, they found raspberries, they found strawberries, and they're like, they live the next day, they're really sweet. That indicates it's good. And the plants have a relationship with us in giving us flavor because they want us to eat them. They want us to eat strawberries whole because then what do? You poop out the seeds, and now there's seeds spread throughout the entire environment that you walk around. So you can see how we have this unique relationship or bond with plants in their flavors, tell us if we can eat them or not, and they benefit that we pass their offspring to different lands that don't have to compete with them, their parents. And so these taste spores are all designed to pick up molecules to detect the flavor profile, and then you, from experience, from luck, in some instances in our ancestors' history, then they send signals and say, oh, this is sweet, this is salty, this is, I'm running out of flavors in my head. What were the other ones? Sweet, salty, bitter, and sour. These are things that I like, these are things that will keep me alive, that flavor my food. And so those are picked up by our taste receptor cells. Remember that the word gustatory means taste. Within them you find basal cells. And what do basal cells, what did they do in smell? The same exact thing. They are the stem cells that will eventually form gustatory receptor cells. And so basal cells will eventually turn into gustatory cells, but I want you to see that there's an intermediate step. The connection basal cells will create gustatory cells, but first they go from basal cells, then they go to transitional cells, and then finally those taste receptor cells. The only thing to add that's a little bit different from the last slide, and you can see all three cells. Transitional cell, and I will add it in order. Basal cell is the stem cell 1. They turn into transitional, and then taste receptor cells 3. The area that gets exposed to the taste molecules, to the food molecules, is going to be your taste pore. And that's all we add here. And so as we look at the eye, some structures that we're going to see when we look physically at the external portion of the eyelashes, which protects matter from going into the eye. The eyelids, which protects the eye when you close it. You have angles to the eye. You have this corner angle and this lateral angle. So they are just named after where they are found. Medial angle or lateral angle. The pupil of the eye. Oh, sorry, let me stay in order. The palpebral fissure is going to be this large groove in between both of the eyelids. The word I got to add here on all of you is that eyelids in Latin, and you should know this, is palpebria. And so you can see palpebral fissure is the gap between the eyelids. The gap between the palpebria. The cornea, if you look at the eye, you can see that there's this white tissue on the outside of the eye. On the next page, you're going to see that this is called sclera, the white portion. But this gets thinner and thinner as it makes its way to that pupil. And then as you see this is the same material as the sclera, but it's thinner. And so you can see through the eyeball. And so you're looking directly inside of the eyeball. This is called the cornea, which is the translucent or the transparent part of the anterior part of the eye, the front of the eye. Say again?
Is cornea and iris the same thing?
They are not the same thing. Cornea is going to be only the cover on top. The iris, everything that you're seeing, the pigmented area, that's underneath. That's like, if you're making a comparison, oh, I have a picture for this. When you look at, say, this photo here, you can see the cornea is going to be the outer covering, and then the iris is completely underneath it. So you're looking inside, and the iris lies underneath. The pupil is the opening in the very dead center that you see here. That's what the pupil is. There's nothing technically in there. It's just empty space, like an opening, much like this. The pupil. And then you can see that the iris, it sits in between that iris and the lens. This huge structure lies behind it, which we're going to see in a second, but it lies behind the pupil, that open space. The lacrimal caruncle. We got to get this word. We know the lacrimal bone was the crying bone, lagrima in Spanish, right? So lacrimal caruncle is going to be this little space that kind of drains tears. Not kind of, it does drain tears. And that's what this is right here. Lacrimal caruncle. And so these are superficial features of the external eye. If we look at the eyeball, we sort of draw it out. What we see is this image. We're sort of looking at it from a lateral perspective. This is the front of the eye. The front of the eye. And then you can see the eyebrows, the palpebria. And now, what's in here? When we look at this region, you can see that the palpebria has this little connective tissue underneath it, underneath the eyelid, that little pink region. And this whole space is called conjunctiva. Conjunctiva, as you're seeing. But you can see that we call the part that's connected to the actual eyelid something, and we call the part that's connected to the the eye, or closer to the bone in a way, another conjunctiva. So the one that's connected to the eyebrow is called palpebral conjunctiva. Nice and easy. And then the one that's connected to the eye is called ocular conjunctiva and also bulbar conjunctiva. I'll put both of these, whichever one sticks best, hold on to that. Ocular is like eye, right? So probably the easiest one. And fornix is going to be this little cavity that they create when they go from being this one to becoming the other one. They are technically the same layer, but we give a different name if it's connected to this or if it's connected to this. Palpebral conjunctiva, ocular conjunctiva. The area, the little space they create is called the fornix. This is the site where you can develop pink eye. What the word pink eye actually is, it's I always have trouble saying this word. Conjunctivitis. I always have trouble saying this word. Never goes away. Conjunctivitis. And then this pink eye, this is going to be inflammation of the conjunctiva, of that area gets inflamed. The epithelium there gets infected. We'll just say it, keep it simple. And so when you have an infection in this area, or the bottom lid, the bottom conjunctiva, this inferior one, again, we call it conjunctivitis. Conjunctivitis. Now, just make sure you know what each one is. And then we have a little gland here called the tarsal gland, and it keeps it lubricated, keeps it moist, prevents the eyelids from sticking. Now, vision, tears. The tears, this part, think of it as a duct system where tears are generated. And so tears are generated in the lacrimal gland, which is always found lateral on the side of your ear. And then it drains in this direction through the tear ducts. Those tears will make their way all the way through at oblique angles and make their way. All the tears that's what keeps your eye lubricated. And when you cry really hard, that's where they come from as well. And then they go into this space here. And in this space, which was your lacrimal caruncle, that was the name of this pink region, you're going to see that you have openings into your nasal cavity. These openings are called the lacrimal punta. You can actually visually see these. If you look inside of the mirror and you look at this region, you'll see two little ducts in there. Those are your lacrimal punta. They're going to send tears through the lacrimal canals, but they're little canals, so we call them lacrimal caniculi, which means little canals. They go into the lacrimal sac, and then they empty into your nose, the nasal lacrimal duct. And then those tears will be drained into that inferior meatus of your nasal cavity. And that's why your nose drips tears when you cry, because the tears make their way into the nasal cavity. And just know this order, that's good enough for this page. And tears are produced in the lacrimal gland. And these tears are there not only for crying and emotions, but to lubricate the eye, nourish the eye, and it even contains oxygen, because there's no blood vessels on the cornea, and so that's how it delivers oxygen through the liquid secreted by your tear ducts, because it doesn't have capillaries on the cornea, which is a pretty interesting fun fact about that. And then it also contains antibiotics. We're just going to put, we're going to have lysosome. It's not an antibiotic, pardon. It is an antibody. Oh, dang, I keep using the wrong tool. Lysozyme, an antibody. And we know what antibodies do. They attack invading pathogens. And that's what your tears do for us.
What happens when you get something stuck in your eye?
Second again?
What happens when you get something stuck in your eye? Like,
Oh,
for example,
so with that, we go to the interior structures. So his question is, what happens when you get stuff stuck inside of your eye? And now this is where your lab list comes into play. So this is going to be kind of where the lab begins and ends. This is where we talk about the lab. And when we look at the eye itself, you're going to see that it has tunics or layers. And these tunics, when we look at the most inner, well, let's start at the outside. The outside is going to be that white layer, the sclera. That's going to be the fibrous layer. That's going to be the white portion that you see. Remember that the cornea is part of the sclera, but it is transparent. A way of learning this easy is that it is a transparent part of sclera. You can think about it like that if you want. They're continuous. As you look at the image, you can even see that they are continuous. Before I get to your question, Miles, when we look at the vascular layer, this layer is going to be the next layer underneath the sclera, which is going to be what we call the vascular layer, or also just called the choroid layer as well. Your list in lab has it as choroid layer. But I like vascular layer because it's telling you what the purpose of it is that it contains blood vessels in that region. And so the choroid in and of itself means vascular layer, which means that means to have capillary network. And we know what capillaries do. They supply oxygen, nutrients to anything that they are near. And so they're supplying it to the neural layer, which is coming up next. And so now this vascular layer has two objects associated with it. If you follow it, you can see sclera, choroid. You follow the choroid, you will see this little smooth muscle region here, which is called the ciliary body. And it controls shape of lens. And then we're going to add plus pupil size.
So the lens changes shape.
Say again?
The lenses change shape.
Yes, it does. Doesn't seem like it could, but it does.
Going in.
Yep. Mm-hmm. It changes shape. Even rotates a little bit too.
Is that how the eye dilates?
Yes. And so to the second part, pupil size, if we look very closely, our pupil is this opening right here where there's nothing. That's a pupil. Wouldn't hurt to label pupil here, but pupil, and then you can see that it is connected to the ciliary body. And so the reason your pupils dilate is two reasons, and not just this, but this is the only, that's kind of where I make a cutoff for our level here. This just controls the pupil size. That dilation, that constriction is somewhat carried out by this muscle. There's another one involved as well called the iris. And this iris is pigmented, controls pupil size 2 plus pigmented. And now before I go into the inner layer, and it also answers Miles' question as to what are floaters. And so when we look at this iris, a fun fact now, not really for the class, but fun fact, what is colored eyes? It's in this iris. Every human being has only one pigment. It's brown. And so when you put brown pigment, it's brown eyes. But then if you take off like half of it, they become hazel. You take off all the brown pigment, then eyes are blue and somewhat green. Blue means less pigment, green means some pigment, hazel means more pigment than green, and then brown is pure brown. So when you look at the iris itself, do I have another picture of that iris? Yeah, this iris, it's always brown pigment, but now again, if you have blue eyes, you don't have it. You have green eyes, you have a little bit. You have hazel eyes, you have a little bit more brown pigment and some areas missing. And then if you have brown eyes, you have brown pigment. I'm sure there have been other instances of different coloration, but most of the time it's diseases or dysfunctional eye. So I've never really heard of anybody that actually has eye colors that could be particularly unique to that. I think that there's like internet rumors, internet memes of purple and gray, but that's not real. Unless gray is real in a disease, red is real in albinoism, I believe.
Is black pigment?
Black, I don't think that. I think black at the end of the day is just
Really dark
extensive brown pigment. Pretty sure. And so, with that,
In biology, they found that the blue eyes were a mutation that happened. So could mutation.
in albinoism, yes.
No, they
Oh,
do that.
but it also happens in albinoism, genes. But
But some mutations could change.
it's the mixture of genes. And yes, the first instance of color in human beings was probably a genetic mutation that didn't affect that person's livelihood. And then they were able to keep passing on their genes. But originally, it's like when you look at all dogs, they all have brown eyes. But I wouldn't be surprised if one day There will be one mutation where a dog comes out with green eyes and then it's bred, and then now all dogs are wanted bred, some of them with dark, some with green. And that's kind of how it kind of originated in humans, just by chance, and then it didn't affect us in reproducing, and then we kept existing and passing down that gene. But originally, yes.
What does it mean when you have two different colored eyes?
I have no idea, and I don't even remember the name of that. I used to know that, and I don't remember. But there is a case where you can be born with two different colored eyes, or just one browner and one lighter brown. I don't remember what that's called, and I don't remember why it happens. But that's something I got to refresh on. Yes, we'll do that during the break. And now, so to finish up here, get our highlights: fibrous layer, sclera, cornea. And the function of those is to surround and protect the eye. It's a site of attachment for the extrinsic muscles, which is a lab topic. Don't worry about it for lecture. We'll end there. And remember, if we can control the lens and the size of the pupil, you are technically able to focus light. You are able.
The arrow to number 2 says last two. Is that what it says?
Say again?
This is an arrow pointing to.
Oh, this
two.
lab topic.
Lab. Oh.
Yes. The extrinsic eye muscles. The vascular layer is going to be the area with a lot of blood vessels, and wherever we have blood vessels, we probably have lymphatics. And since it's connected to smooth muscles, you can see where this comes from. So that includes iris, ciliary body, and the choroid. That constricting and dilating of the pupil regulates the amount of light going in. We know it's vascular, route for blood vessels, lymphatics. Controls shape of lens, which help with focus. And then there's a fluid in this area nutriating all of this as well, called aqueous humor. That's the fluid inside anterior chamber. And we've made it to the retina, the most inner layer. And this retina is the site where you are going to have your photoreceptors. This is the site of the cells that are sensitive to light. So as we look at this, and don't forget your lenses here, but then as light goes in, it goes, and then it'll bounce, and then whatever you're seeing will be projected onto this layer, which is the retina. And then look how convenient this is for us, the optic nerve, cranial nerve number 2. You're looking at it right here. All of these retina cells are going to pile up their action potentials of what they're seeing, and then they're going to send in the axons containing all the information of what you're seeing, and they're eventually going to go to what lobe is going to receive visual information? Close, starts with an O. Occipital. So all of this will eventually be sent to the occipital lobe, where you will process it and interpret it. And in your eye, I guess that's it for the occipital muscles. Nice. What to take from here? Do you have ora serrata? You don't have it on your list. And so what I will say is that you only need to know 2 things, and they're right here: the anterior cavity and the posterior cavity. That anterior cavity, as I mentioned before, this space has aqueous humor. Within this space, within this anterior cavity, you have two chambers, an anterior chamber and a posterior chamber. Oh, sorry, right here, posterior chamber. So anterior chamber, and here we're gonna write posterior chamber. I don't want you to write as noisy as I am, but I want to be clear that it's this whole area here. And again, it circulates aqueous humor. You can see that fluid is circulating here, and then it keeps the eye nutriated, contains gases, nutriates these cells, things like that, keeps it moist. And then we finish up with the eye on the posterior chamber, and to finish up with Miles' question, is that in this posterior chamber, dang it, you have this large gel-like substance in the back here, in this region in here, and this gel-like object, oops, I traced it wrong. This gel-like object is called vitreous humor, and it is what makes this gel-like, this gelatinous object, which means gel-like. And so inside of this vitreous body, which contains vitreous humor, it's a little sac. I'm going to write it simple. Sac, gel-like. I want you to think about what would happen if I were to put a pen in Jell-O and put it to make it in the fridge. That if I were to put this pen in the middle of the, like just put it in the Jell-O, and then I put it in the fridge and I take out that Jell-O, that pen would be stuck in there. That's what floaters are. When you look at the sky, right, and there's always that little thing that you're trying to look at it, you can never really catch it. And that's because your eye, if your eye is circular and made out of gel on the inside, then if you're stuck, and when you turn your eye, it goes with it. It's like embedded inside of that vitreous humor. And so no matter how you turn your eye, like that little object is just right here. Let me draw it large here. It's just right here. So no matter what, when you look at, as you move your eye, even if you move it that way, this will shift this way because your eye moves as one. And so they are little pieces of your retina that have gotten loose and now they're stuck inside of that gel. And that's what floaters are, is that you have little dead cell gunk grouped up and piling up within your gel. And it gets worse as you get older, but then it clears up and you can get you can get it cleared up with surgery. Now, that's all the posterior cavity has, and we're going to end it with that for this page, because I am making some cutoffs. And then the only last thing is make sure we know the retina contains photoreceptors. You're done with this page. Don't forget that your eyeballs are connected to optic nerve number 2. We know the function, therefore, optic nerve is involved in vision. Only thing to add. And then let's finish up with the ear.
There's nothing else to do. Is there anything you don't want us to learn?
Nope, that's absolutely it. And then we finish up with our ear and finish up our lab material. And so you'll see that there was a model for the ear, and this comes up with our last term. So I'm going to stay focused on what your lab list has, and that's what we're going to work on for the lecture as well. And so we start out with the outside of the ear, which is the external ear. You have your auricle, which is a satellite that's but it actually concentrates sound. And then those sound waves will travel through the external acoustic meatus, which is the opening in the skull that you learned about in the skeletal system. We're then going to hit the eardrum in anatomy. This is referred to as the tympanic membrane. Then it's going to vibrate your malleus, vibrate your incus, vibrate your stapes. These 3 bones are your ossicles, and they are in that order, M-I-S. Malleus, incus, and stapes. I always think like when little kids are talking to their teacher, they always say Miss, and I always remember it. Miss, M-I-S. Malleus, incus, and stapes. So sound, like the letter A, if I say the sound, it's vibrating this in a way. If I say O sound, it's vibrating it in an O sound. And then it relays those vibrations, whether it's A or O, or into the same area, into this labyrinth. This region, as you look at it, you need to see that there are two halves, this half and this half. This side cares about sound and listening. This other side cares about balance. And so when we look at, before we get into what this apparatus is, you'll see that sound as it vibrates here, it has to vibrate, has to be interpreted, and then that sound has to escape. And look at what it uses. It uses a tube that's connected to the pharynx, to the nasopharynx. This is called your auditory tube. Your ear is connected to your pharynx. That's why if you get an ear infection or a throat infection, a lot of times what will follow is a follow-up ear infection. And that's because it makes its way through the auditory tube. But in nature, this is designed to just allow sound to sort of escape out, because sound is a force. And so if it went in and has nowhere to go, it's just going to collide against this, and believe it or not, that would do damage. And so this leads sound out. It leads out through your nasal cavity and through your mouth, sort of allows sound to escape.
What does it mean when your ear pops? Like, if you're
That's
like,
usually probably in the middle ear pressure, that you can have pressure built up, or there's like mucus completely plugged up here. And then if you you do that thing where you breathe in, or you go to different levels of the atmosphere, like different elevation, then you'll see that pressure might cause like this, like air is getting built up, and then it just powers through and sort of makes it smaller and sort of allows it to go through. And so that's kind of what the feeling of pressure popping in your ear is. And then this segment we just described is your middle ear. As we look at the inside, you'll see that we have what is called the labyrinth. This whole thing is called the bony labyrinth. It's made out of, in an essence, calcified tissue, bone. And that's all there really is to add. Be able to label these. The auditory ossicles, know them. Again, I guess. Remember the order. Know the terms in bold. That's all it really is knowing what the terms in bold are. Be able to describe them with one sentence, and it's right after the bolded term. And remember, elastic cartilage found in the ear. The auricle is made out of elastic cartilage. And then hanging in there, we got the last little bit, which is just this page to end the class. This is more on the same page. Now, what exactly is in this bony labyrinth? This bony labyrinth, first, what are the regions to it? You have the cochlea, vestibule, and semicircular canals. The cochlea is going to be this spirally portion responsible for sound, recognizing sound, hearing. This other region, the vestibule and the semicircular canals, these are responsible for balance.
What's it called when you can't balance? Like litigo? No.
Vertigo.
Vertigo.
And so how you detect balance is that if you look at the vestibule and you look at the semicircular canals and you look at these regions, what you see inside, especially inside of these semicircular canals, what you find is this. Inside of these three regions here, you encounter what's called the cupula. And this cupula, what it is, it's sort of like a gel-like substance, and you have hair cells. And so these hair cells, what they do is that as you move your head around, your body recognizes the movement of this object around the hair cells, and they send the signal, hey, my head's tilted this way. If it's straight, it's just normal. You feel normal. But the second that you tilt your head sideways, this cupula knows that if you have issues with this, your head could be like this, and you probably think you're like this. Right? And then believe it or not, like your eyes would adapt that if you kept your head like this forever, that no matter what you see, your visual image, even though it would be like this, you would all technically be slanted, but it would look like this to me. So I would never know if that my head was slanted if I didn't have this sort of bent down. So this gives you a feeling that your head is bent. If you don't have that functional, your head could be like this and you think you're like this. Because your eyes will adjust. Your eyes will make it look like it's normal. But if you don't have this to detect head movement, you will not be able to detect that your head is in motion. And that's because, again, it's just the positioning of that cupula determines what your body senses. Is it bent over? Is it spinning? Etc. And that's what the semicirculars do. When you go to the utricle and saccule, it's more of the same. If you look here in the vestibule, the part responsible for balance, if you look at the center part, there is these two regions. If you zoom in even more, you would see more of the same thing. You would see hair cells. These hair cells are in contact with a gel-like membrane. And inside of that gel-like membrane, you create little inorganic crystals. You create a salt in your body, an ion, little crystals called otolith. And so these otolith crystals, they're in this gel. And so as this part of your balance, what it recognizes is acceleration and deceleration. How? Look at these hairs and make an image. When you teach somebody new to drive, I don't know if you've had that experience, but if you're teach someone to drive and they press the brake for the first time, they always press it too hard. And what does your body do?
Sure.
Like this. That's what these hairs do. When you're running really fast, these hairs are like this. And because the gel is like going back, and then the hairs are like, ugh. And then you stop, and then those hairs go forward, and then they detect how much fast you're stopping. So these hairs, if they're like this, you can feel how fast you're going. You've all had it in your life that you're running, and you know, okay, I'm running really fast. If I fall, I'm going to get hurt. You can tell how fast you're running because of this. This will be sort of putting pressure on it and going in this direction. Let me do it for the slides. Here, going in for the video, going in this direction. And then when you stop, those hairs kind of feel all of this just go in this direction when you stop. And so this is your mechanism by which you detect what we call static equilibrium. And this is a fancy way to say acceleration and deceleration. Acceleration and deceleration. Static equilibrium. This particular object is found in the vestibule. The vestibule contains the utricle and saccule. And the utricle and saccule contain hair cells. And they detect the movement of ear stones.
They're just like always there.
Yes, always there.
Are you like born with them?
We're born with they develop a little bit, they mature, right? When a baby can't necessarily run right away, but they're born to build it. Well, as soon as we start walking and running, they get a little more refined, right? If you're into like extreme sports, yours get a little bit more refined. Like if you're a gymnast and you're doing tricks back and forth and doing flips, like in the beginning, like that could be pretty exhausting. At least for like in the dance world, at least in the thing that I do, spinning a lot, like to teaching somebody to do like one spin, like then you kind of just see them like, whoa, like, like it's, but then like after a long time, like you're just spinning, and then you kind of don't even like feel dizzy. Like I can do this for hours and hours, but in the beginning, getting somebody to do the first one, they're like, and then, it's just, in the beginning, that's probably how I look too, and then, but that's trained better with time. And so that can be done both the forward acceleration, like race car drivers probably have a defined one, skiers, things like that. And so gymnasts and anything that requires a lot of, agility is probably semicircular canals. We call this type of control, balance control, we call it dynamic equilibrium. And that means crazy rotational movements, like dynamic movement. And that is the end of this section.
Can you go back to the other page?
Which one? This one back here?
No, the last one.
Oh, yes, this page.